Thank you very much. Gabe, can you confirm that you hear me?
We can.
Thank you very much. A very big welcome to everyone to this presentation of our virtual seminar series. This is a pharmacometric discourse, clinical applications and research. Just to remind everyone, the goal of this seminar series is to share knowledge, share experiences, best practices, and locally relevant solutions. This seminar series is organized by Pharmacometrics Africa, the Ugandan chapter. And today we are really privileged to be hosting Karen Yo. Karen has been working in the field of PBPK for over 20 years now. She has likely been working in aspects of drug development. Recently, she's been working in the field of global health, something that she is hugely passionate about. I cannot imagine any better speaker to take us through the application of PBPK than Karen. I would like to call upon all the participants to deeply engage in this presentation. We have the Q&A tab at the bottom of your Zoom. Please type your questions there. If you've seen a question that resonates with you, please emphasize that question so that it can be robustly discussed. We're very happy to hear from you, Karen. Over to you. Thanks very much.
Okay, thank you very much. First of all, I just want to make sure that you can hear me and see my slides.
Yes.
Yes, we do.
Okay, perfect. That's great. Okay, well, first of all, thank you very much for the kind introduction and also the invitation to speak. For those of you who know me, you know that I'm very passionate about talking about PBPK. It's always an absolute pleasure and a privilege. So before I get on to talking about the moxidectin case study, which is interesting itself, I just wanted to give you a little bit of background about how I got into this area, because I think for those of you who are early career, individuals, sometimes you just don't know where you will end up in your career, and so I'd just like to spend a few slides talking about my story, and then talking more about PBPK before I actually get onto the moxidectin case study. So I was actually, so I've actually spent most of, my formative years in growing up in Africa, and I lived in Zambia, I lived in Liberia, I lived in the Democratic Republic of Congo. Also Kenya and Tanzania before finally settling in Zimbabwe, and then I went to university in South Africa. And I think it's always struck me when you talk about Africa, I think most people think that the things that you have to concern yourself with are the large animals, or the like elephants and crocodiles and things. And I think that many people are unaware that often it's the things that you can't see, or the tiny things, that can actually present, quite significant problems and challenges. I know myself when I was growing up that, we had malaria. I remember growing up and I was covered in sticking plasters because we had putsy fly, and schistosomiasis, bilharzia. But, the one thing that I wanted to mention is that, we lived in very remote areas, often on the borders, and we didn't have great healthcare. And as a child growing up, I remember taking large doses of medicine. And at the time, I didn't know what it was, but I just knew that they made me feel really awful. But obviously, helped me to recover from whatever, disease or problem we had. So, and then after, growing up in Zimbabwe, I went to South Africa to Pietermaritzburg University. And I loved maths and I loved physics, and I wasn't sure what I was going to do with it, but I started on a course there. But while I was at university, I also took chemistry and biology. And in fact, met Professor Drivers there, who was very passionate about medicinal chemistry. And it was while I was studying with him that I picked up my interest in medicinal chemistry. And what we used to do was often go, as a class, we'd go into the bush, we'd bring back, large amounts of, endogenous plants. And what we would try and do is extract, what were thought to be active ingredients. And so I had this huge passion back then, but I didn't really know where I was going to end up. I love medicinal chemistry. I love extracting active moieties and then trying to figure out how we would then actually get back to, forming these products. But also I kept up this interest in maths and physics as well. So when I left university, just before I moved away from Pietermaritzburg, I spent about a year working in the laboratory trying to get enough money together to get a plane ticket to come to England. And when I got to England, again, I didn't know what I was going to do. I sent my CV around to various universities trying to get a PhD position. And Professor Tucker invited me, to do a PhD with him. But at that point, it was in drug metabolism. So I'd got physics and maths behind me, organic chemistry, but I hadn't really done anything along the lines of drug metabolism. I ended up doing a PhD in CYP2D6 mediated metabolism, followed by a postdoc in childhood acute lymphoblastic leukemia, focusing on 6-thioguanine nucleotides as a treatment. But while there, I continued to, enjoy working in this area as well, and ended up joining a clinical pharmacology and therapeutics group where my main focus was actually cardiovascular risk assessment, looking at the effects of smoking, high blood pressure, cholesterol, and statins. And because of the number of co-prescribed drugs that were given with cholesterol, I soon developed this interest in drug-drug interactions. And in fact, really, this is where I started, my publication and my interest in assessing drug-drug interactions. And it was at that point that while I was doing a lectureship in clinical pharmacology and therapeutics, Jeff Tucker and Amin Rastami set up SimSip and asked me to join them. And this was really where my interest in PBPK modeling started. Now, PBPK modeling has come a long way. I've been working in the area for 20 odd years. And really, it's had a significant impact on the drug label and specific with respect to drug-drug interactions. And this is just some of the therapeutic areas where it's had an impact and some of the drugs as well, where PBPK modeling has actually been used to inform the drug label. And this is why it's really taken off, and it's because of the ability of PBPK modeling, I think, to actually replace clinical studies, and for pharmaceutical companies. So this has really been a big area over the past few years. This is a publication that came out very recently that I was a co-author on. And what we're trying to show here is dose optimization informed by PBPK modeling. And you can see that in 2013, which is, just a little over 10 years ago, there weren't actually many examples of where PBPK modeling had been used to inform the drug label. But you can see now in 2023, the cumulative number, it really is significant. And with respect to DDIs or drug-drug interactions, it's had a significant impact, not just with CYP-mediated DDIs, but also transporter-mediated, proton pump inhibitor effects. Also, it's been used to understand complex absorption and food effects, and also look at specific populations as well. So it really has taken off. And when you look at the different therapeutics areas, you can see that, not surprisingly, because of the number of drugs that have been developed in the oncology area, this still leads the path or the way forward in which PBPK modeling has been used to inform. But I just wanted to mention this publication because even though we're talking about DDIs, what we're talking about as well is the way that the regulators think and about the way that they actually assess the model and how progressive it's been, especially with specific populations and assessing the dose that is required. So why is there this significant interest in PBPK modeling? And I'm going to take you back to basics here and talk about the individual components. So when a drug is being developed, a lot of data is collected on it with respect to absorption, distribution, metabolism, and excretion. All of this in vitro data that then gets assessed as it goes through regulatory review. What we can do with PBPK modeling is take that in vitro data, scale it according to the organs, and then feed it into a series of differential equations which actually describe the physiological processes in the body. When we combine this with trial design, what we can do then is actually predict the PK parameters, so we can predict the clearance and the volume, but also we can actually look at what the concentration time profile looks like. And so we can do this before we have any clinical data. And often PBPK modeling, it starts being used in drug discovery when you collect this data, and then we can predict PK parameters and a PK profile. And once we've actually collected that clinical data, what we can do is overlay the dots and see how well the profile and the PK parameters actually predict. Now, obviously, this is ideally what we would like to have, a good match between the predicted profile and the clinical profile. But as we know, sometimes with modeling, if we don't have a well-described model, then essentially we can end up with a misprediction where we may have a lower Cmax, we may have a longer half-life, and therefore what we would do is go back to the in vitro parameters or the original model and have a look to see whether there are any missing components or some parameters that actually need to be optimized in order to be able to capture this profile. So this is really what we're talking about with PBPK model. It's taking drug-related data, combining it with system parameters and predicting this profile here. Now, this is of interest to us because what it means is that once we've got a baseline model that is working reasonably well in healthy volunteers, we can actually then predict in another population if we have the relevant system parameters. And so what I'm going to focus on here are the system parameters. And if you think about this, essentially what we need to do is we need these distributions for different organ sizes. So if you think back to the previous slide, we've got this physiological model where we have different blood flows, different organs. And so what we do is we collect these data from the literature and actually fit a distribution to this. And then what we can do is when we're building these virtual individuals, we assign a real organ size or real blood flow from this actual distribution based on real data. But it's important that when we do that what we need to do is make sure that the values that we're actually generating are reflective of real data and real individuals. And so where possible, we try and build in this covariate relationship. So look at the age, gender, also the impact of ethnicity and disease. And so we're trying to build individuals that actually reflect the real life scenario. And of course, what we do is we use a correlated Monte Carlo approach to capture individuals with appropriate size organs, as we can see on the right-hand side here. So once we've done that, we can run a simulation. We've got this virtual population. And what we would do is we'd end up with a range of AUC values or predicted drug-drug interactions for these virtual subjects, which is reflective of a real population. And therefore, what we can do is actually go back and have a look at these individuals and their baseline characteristics and see why one or the other has got a higher or a lower AUC value. So that's just talking about the basics of PBPK modeling. If we want to go ahead and make clinical studies, more diverse and more inclusive, or look at different specific populations, then what we need to do is is collect the data for these other populations. So you can see that there are cancer populations, organ impairment populations, also an obesity population and a specific NASH population. We can have a look at the gender effects, ethnicity effects, and also effects that reflect changing age or changing physiology, perhaps in children or pregnancy and also lactation as well. But I think the key one of the important things to mention here is that many people sort of think that obviously for PBPK modeling, it is very data driven and you need a lot of data. And that is the case. But often when we're looking at specific populations, it may be that there are key parameters that we can focus on that we know change. And just by having those alone in the simulations, we can actually capture what is seen clinically. So if we have a look at this diagram here, what I'm talking about is on the right-hand side, we're saying that, say, for example, you've developed a compound for a particular drug. And by that, I mean what you've done is you've collected the ADME data, we feed it into the PBPK model with the differential equations, and what we're predicting is the PK in adults. But if we want to know now, how do extrinsic factors like smoking, diet, or alcohol, or different races, or different genders, or age-related factors, what actually happens then for the PBPK model? We take our base PBPK model that we've developed in healthy adults, then we apply the new population. In this case, let's say we're talking about children. And what happens then is the system parameters for children now replace those for adult, and we can predict the PK in children. And in fact, a nice example here is deflazacort, which is actually used to treat Duchenne's muscular dystrophy. And this is an example here where a model was developed for the drug in adults, used to simulate the drug-drug interactions, and then applied to appropriate doses in children. So this is just an example of what we would actually do when looking in children or applying a specific PBPK model in drug development, or in fact in global health scenarios. So an area that has actually generated a lot of interest recently is predicting exposures as a consequence of pregnancy, or indeed through lactation. So in other words, what are the predicted milk to plasma concentrations? So if you have a look at this diagram here, you can see that this is typically how we would represent a PBPK model here by the different organs and flows. If you have a mother now taking medication, what we can do is simulate the plasma exposures of mothers and then use physicochemical parameters to actually predict the milk parameters, and therefore convert this to an actual milk to plasma concentration. So what we end up doing here is we end up with a profile for the mother in the plasma, a profile in the milk for the mother based on predicted milk to plasma ratios. And then we can use this information to actually calculate an infant daily dose and a relative infant daily, and relative infant dose. And in fact, this is typically the information that we would want to extract if we'd actually conducted a clinical lactation study. It would be how much of the drug actually ends up in the milk, what does the profile look like, what is the infant daily dose, and what is the relative infant dose. But what we want to do, and what we've been doing, and in fact, what we're going to showcase with the moxidectin sample, is we actually want to move things beyond this. What we want to do is to take this information and say, okay, so this is the amount of the drug that actually gets transferred into the milk. What would the infant daily dose be? And we can actually run simulations in breastfeeding infants to predict what the exposure is actually going to be. So in other words, rather than just predicting what the milk to plasma ratio is going to be and the exposure in the milk, we're actually running simulations in the breastfed infant to predict and simulate the actual exposures. So now I'm going to move on to this moxidectin example here, and where we actually, this was actually a drug that was developed for the treatment of onchocerciasis, which is river blindness. And this drug was approved for use by the FDA in 2018 in patients aged 12 and older. And when making recommendations about, mothers and, and their breastfeeding infants, obviously it's always a balance between efficacy and safety. We want the mothers to be treated, but we also want the breastfeeding infants to be safe. And therefore, for this particular example, at the time, this was the labeling language that was actually presented when for the original submission to the FDA. And you can see here that a clinical lactation study was actually conducted. Moxidectin was detected in the lactating women following a single 8 milligram dose of moxidectin. And but you can see that really no specific guidance was provided with respect to dosing recommendations for breastfeeding mothers. And this is not surprising. In fact, I think it's quite typical of many of the labels that you look at, specifically for breastfeeding mothers. If clinical lactation studies have been conducted, it's reported. There may also be toxicokinetic data conducted in animals that are also presented, but often, as is the case, no specific guidance regarding dose recommendations is actually made, and therefore it's left to the healthcare professional to make decisions. So now I'm going to talk about this publication here and what we did at the time to address this. And this is a publication that came out very recently. These are all the authors that were involved in this drug. So it's moxidectin. And what we used were quantitative clinical pharmacology approaches to support moxidectin dosing recommendations in lactation. So in other words, what we wanted to do was take it that one step further and try and give as much information or utilize the data that were available and into making a more informed decision. So since the initial approval of moxidectin, additional non-clinical toxicology data were generated, and it was hoped that these could actually inform maternal fetal risk. So these data were used in conjunction with the existing clinical data, and what the objective was to establish the level of exposure of infants to moxidectin via breast milk, with a view to helping prescribers and policymakers make informed decisions for its use in breastfeeding. So, making an informed decision, what information do we actually have available? So moxidectin was detected in the milk of lactating women following a single 8 mg dose. In a pre-postnatal study in rats during the lactation period, there were clinical signs of showing, including weight loss, but also increased mortality in the rat pups was actually reported. And so at the time, reflecting on this and about the best way forward, we were discussing, should the advice to women be to abstain from breastfeeding at the time of treatment with moxidectin and for about 6 months of treatment? Well, moxidectin is actually given as a single dose, but it's got a very long half-life and stays around in the system for quite a while, for about 6 months at low levels, but it's still there in the actual system. And so this is something that you have to consider, especially when making this decision. So this is what the clinical study exposure data looked like. And you can see here on the left-hand side, we've got a plot there of showing the exposure in plasma and also the exposure in breast milk. And you can see that very intensive sampling. I mean, I don't know, I've looked at many studies over the years, or certainly those that have been conducted, and not seen sampling like this. But obviously you can see that for this drug, you've got a very rapid absorption phase followed by an extensive or half-life and elimination phase as well, hence the extent of the sampling. And on the right-hand side, you can see here that we've actually got the parameters reflecting the breast milk and also plasma data. And as I mentioned, one of the things that we often report out for clinical lactation studies is this relative infant dose. And you can see here that it's about 8.7%. And this is below the WHO recommended RID safety threshold of about 10%. And therefore, typically we can look at this and think, great, but in terms of actually helping us to make dosing decisions by, for moxidectin, certainly in my view, it doesn't really help. But I think the other thing to say here is that obviously we've got this, WHO recommended safety factor in the fact that it's below that. But when you actually have a look at the profile of the drug, you can see one of the concerns is that you've got this very rapid absorption phase here, where you've got quite a high Cmax before it actually drops out over the week period, and therefore this is something that we wanted to look at in the modeling. So when doing the modeling, what we have to think about is, what do we actually use for the dose? Because do we actually use an average dose, because we're talking about a single dose of the moxidectin here, or do we use a maximum concentration? And this is the way that we would typically calculate this. Infant daily dose equals drug concentration in milk times by the daily volume of the milk ingested. And this is explained very nicely in the citation indicated here. If we look at the moxidectin example, you can see that the average milk concentration, we end up with a mean infant daily dose of about 1.1 microgram per kg. But if we use the maximum milk concentration, then we're using an infant daily dose of about 23 micrograms per kg. So they're going to give you very, very different answers depending on which one you actually use. So what we ended up doing was using the clinical lactation data to calculate the amount of moxidectin actually excreted in breast milk each day over the first 7 days. And what we wanted to do as well is have a look at the inter-individual variability. So we're saying on average about 20 on the first day, that's micrograms. But in actual fact, you can see that the spread ranges from about 5 up to about 45 micrograms. And that's something that we can actually address in the simulations. So with respect to the PBPK modeling itself, obviously in terms of the actual dose that's given, we've got variability and we've got different things to consider there. But when you think about moxidectin itself, it's a CYP3A4 substrate, undergoes some biliary clearance. The C log P value is reported out at about 4 to 5, but log P is was actually measured to be greater than 6. It is very highly lipophilic. It's highly permeable, and it's got a very low fraction unbound. What we can do with the PBPK model is actually look at the distribution across tissues. So far, when I've talked about PBPK modeling, what I've mainly talked about is actually predicting plasma concentrations, because really that's what we use to actually verify the model. But the beauty about PBPK is we can actually predict concentrations throughout the different tissues. And for a drug like moxidectin that's highly lipophilic, it's got extensive tissue distribution, and this is something that we could actually look at with the PBPK model. But it's important to recognize that in these neonates, we need to be looking at the ontogenies, and specifically with respect to moxidectin, the CYP3A4 ontogeny, biliary clearance, and also the plasma protein binding itself. The fact that it's extensively bound means that the ontogeny of the protein binding, of the plasma protein, could actually have a significant impact. And this is a very busy slide, but I just wanted to show you that within the PBPK model, what we can do is have a look at the physiological changes as a consequence of age. So in other words, if you think about an infant that is breastfeeding, from when it's born up until 2 years, what we need to be able to do is actually factor in these different changes. So in day 1, the infant's going to already have changed, when we get to day 7 and day 30. And therefore, we have to be able to build in these factors, especially if we're wanting to look at the change in exposure as a consequence of age and also breastfeeding as well. And these are just the different ontogenies, some of the different ontogenies that we can consider. And of course, this was actually factored into these simulations. So as I mentioned previously, before we go on and have a look at predicting in children, what we need to be able to do is actually have a look at the model and see how well it's predicting in adults. In this particular case, on the left-hand side, we're showing males, and on the right-hand side, we're showing the women who were actually part of the clinical lactation study. So we're just showing here the performance of the model. And I just want to point out that this was actually quite an early model, and we do have more complex models that have been developed since then that take into account all the food effects. But for the model that was actually used for this purpose, what we're showing here is the actual model verification in terms of capturing the adult data. What we did then in terms of PBPK simulations was what we wanted to do was look at worst case scenario here. And what we're saying is that we've calculated what the amount of moxidectin that was actually excreted into breast milk each day over those first 7 days. And what we're saying here is, let's assume that that full dose is taken in one feed. Of course, it's not practical, but what we're doing here is looking at the worst case scenario and saying, even if that entire dose was given in one feed, we're saying that the relative difference in exposure is quite significant. So the adult here is shown in black, and obviously it's a single dose of moxidectin that's given. And what we're showing here is breastfeeding over these first 7 days, taking the doses that we actually estimated from the breast milk profile. And we're showing that even if it was all consumed in one feed, the profile and the exposures are still significantly lower than we would actually see in the mothers. And what we were able to do was simulate various different scenarios, look at different ontogenies, and also changes in the protein binding as well throughout this 7-day period. Importantly, and this is a figure that actually appears in the paper, what we're showing here is that at the top of this figure, what we've got is the rat pup exposures based on the maximum dose, and we've got rat pup exposures here at a lower dose. We're showing the dose here, or the plasma exposures, and also the milk exposures in the mothers who were recruited in the clinical lactation study. And below, we're showing the PBPK predicted steady state simulations at 0 to 1 month age and from 1 to 6 months age. So what we're saying here is that even during that first day 1 to 7 period, and also subsequent right up until 6 months, we're getting much, much lower exposures than we would expect to see in the adults, and we're certainly expected to see in the rat pup data. So these were information appear in part of the paper, and were used as part of a bigger ClinPharm MIDD strategy to actually inform dosing. So the label has been updated since then, where you can see here that breastfeeding is not recommended during treatment for the first 7 days after treatment. And of course, that's because there remain concerns about the fact that you still have these relatively high doses in the milk, but certainly post 7 days, we were confident enough in the simulations that we're able to actually continue have the recommendation to continue breastfeeding after 7 days of treatment. But it is, we hope that certainly now this publication's out, that it can also help provide guidance for prescribers and policymakers in making appropriate decisions for the use of moxidectin in breastfeeding women going forward. So what I wanted to do now was finish off just with, my last slide and talk about this a little bit more. So that was an example of moxidectin. What I wanted to say is that there are many models out there, and we can use these models to actually apply and assess and predict the exposures in breastfeeding infants. And in fact, this is what we did for efavirenz. What we did is there's this complex model already available in the literature. We utilized this model. We're considering CYP2B6 mediated autoinduction, CYP3A4 mediated autoinduction as well. and this complex process. And we're looking at this in the mothers according to different CYP2B6 genotypes. And what we can do is predict the exposures in breastfeeding infants according to different genotypes as well. And these are just the observed versus predicted data, but you can see this in more detail in the reference actually cited below. But the point I'm trying to make is that what we're doing here is we're actually using PBPK modeling not to replace clinical lactation studies, but to support them. Because when you think about the clinical studies, they're often conducted in a small number of people. And then even afterwards, there can be all kinds of questions about, when the sampling was done, whether the impact of fore versus hind milk, the hematocrit content, would that affect the milk to plasma ratios? So if we can develop a model, we can use it to support clinical lactation data. And importantly, what clinical lactation studies don't often do is measure the exposures in neonates or breastfeeding infants. And this is something that we can actually estimate using the PBPK model. So this is an example here showing the fact that we're taking into account the time-varying physiology, the different ontogenies, and importantly, the different genotypes. But also, there are many models available either for compounds in development, but also global health molecules as well. And in this publication here, what we're showing is a series of PBK models that have been actually developed to support anti-malarial drug development. And in fact, we've used a number of these models, try and, support clinical lactation data and to help, progress this area in terms of providing more information for prescribers. So with that, I'd like to thank you for your attention, and I'm very happy to take any questions that you may have.
Thank you very much, Karen. Such a wonderful and clear presentation. We have quite an interested audience here. I've noticed that there are also quite some people who already have a bit of experience on PBPK. I've seen Andrew on the call. I've seen Shakia on the call. I've seen Kenta on the call. So thank you very much for joining. And I really hope that the audience does benefit a lot from your expertise. There's one specific question on the chat. What I'm going to do is I'll first put aside the bits that I have, and then I'll go into the chat. Karen, you started us off quite nicely how you ended up doing PBPK. And I think this is quite interesting for a number of the junior scientists that be on the call and they perhaps want to get a grounding on PBPK. If I get it right, you didn't actually do PBPK as part of your doctoral work, but you rather went into it after you had done your doctoral work in drug metabolism and the likes. What was your experience really going the direction of quantitative pharmacology after having done, your doctoral work in something that is not exactly quantitative pharmacology? And I'm asking this because perhaps there could be a pharmacologist on the call and they're thinking, can I move into this career path given that I've not done this as a formal doctoral training? The second question, a more general question, the second general question that I'll put across and then maybe for now stop there is, with moxidectin, the example you've given, I know there are a lot of drugs that have been studied in, well, not so many, but there are some drugs where there's evidence that they distribute into breast milk. How exactly did moxidectin end up being in this privileged position of being studied up to actually changing the label? How did that process begin? Was it some request from the FDA? Where did this motivation to study to this point come from? Thank you.
So, sorry, I think there were two questions there. The first one was more about how, I think, how do you end up in a quantitative area if you're more of a clinical pharmacologist? Is that correct?
Yes, you did formally studied quantitative. Yeah. Go ahead, please.
Yeah, so I think, that's, to be honest with you, that's why I mentioned my career path really, because I think, it can go one of two ways. I think, from my, based on my background, I obviously ended up in I was started in a quantitative area, but really, I'm more interested in the application of it and how we can use it to actually inform better use of medicines. And I think I was probably lucky in that I ended up working in a department where there was drug metabolism, but also clinical pharmacologists who were very interested in the use of quantitative medicine. And so, say, for example, I was working in an area of cardiovascular risk assessment where you automatically using data and algorithms. So what we would do there is we would actually use data that were available, create virtual individuals, calculate a cardiovascular risk assessment, and then work out the proportion of the population that, with a certain amount of risk. So I think really that's, where my interest stemmed from. I started off in a quantitative area, but ended up being in a clinical pharmacology environment where people were interested in the use of quantitative medicine. So that really helped me somewhat. And to be frank, if I hadn't ended up there, I don't really know how I would have ended up using my quantitative background unless I'd ended up in industry, say, for example, where, obviously MIDD approaches are used quite a lot. So I think that's the answer to that question. And I think the reverse side of that is that if you're a clinical pharmacologist and are more interested in MIDD approaches, I think that it's certainly happening more routinely now. I think MIDD approaches are being utilized more as part of clinical pharmacology. And therefore, I think it's probably easier to actually, get involved and do it that way around. The answer to the second question regarding the impact on the label and moxidectin itself is that this was obviously the moxidectin MDGH went to the FDA for approval for the drug, and a pop PK model was available and submitted at the time. But obviously, as you start looking at different populations and children, and and trying to make more informed decisions about the dose, this question came up about the best way forward. Obviously, the 6-month, breastfeeding gap or no medication, that's not an ideal situation. And therefore, at the time, what we did was we were looking at a PBPK model and thought, well, PBPK models are ideally suited for this, especially in children under the age of 2. And therefore, what we'll do is we'll develop a model, have a look at this, and then submit the data and then see, how things go. And so, all of this work is described in the publication, but that's really, it was an MIDD informed approach that actually led to this, and PBPK was part of it.
Thank you, Karen. I'm not sure if you have access to the chat. There are quite a number of questions actually your side. Otherwise, I can read the questions for you if you have access to the question.
Yes, platform. I do. Yes, so
yes.
So I'm looking at the first one, which talks about model validation and the criteria of observed predicted ratios between case by case, such as how to select this has been obviously of significant interest, over the years, I think, with respect to the regulators about what is acceptable. I've been involved in the development of many different models, and and I think 0.5 to 2 tends to be more acceptable. In fact, often 3 is used when you're in a discovery space. But I think when you're looking at making, recommendations for dose in specific populations, I think we need to be a bit tighter than that. But again, it depends on the therapeutic window as well. I tend to, it depends on first of all, it depends on the amount of data that you've got available. But typically the way that we develop the models, we would develop them in healthy adults and then hopefully have some drug interaction data available that would help us to refine the individual enzyme components. And as more data become available, they can actually be used to inform the model. And therefore, when we're looking at the rigor of the model, we tend to start looking at 0.8 and 1.25 as the bands. But again, as I said, this has to be put into context of the actual therapeutic window, because it may be that we can look at a broader range if we've got a much broader therapeutic window.
Yes, thank you, Karen. And I think that you can confirm whether this adequately responds to the question. The next, I think, Rim is saying, interesting, thank you. We have quite a number of questions coming in from Henry. I think in the interest of having the discussion within the next 10 minutes, we'll select a few of these questions and then we can follow up the discussion out of the presentation. So Karen, Henry is asking a couple of questions here. And the first one is really around parameterization of PK models, especially in the context of long-term pharmacokinetics. What are the considerations you make in parameterizing PBPK models for scenarios of long-term pharmacokinetics, particularly in adipose tissues?
Yeah, this is quite an interesting question, because obviously moxidectin itself, there were actually some tissue biopsy data available and showing the exposure of the drug in adipose tissue, and obviously it's selectively it basically selectively transfers there, and the partition coefficient for adipose is actually quite high. I think it's about 65-fold, relative to actual plasma concentrations. And this is something that we can actually model. I mentioned during the presentation that this initial model that has actually been presented in the publication, it was very much a preliminary model, because we were actually at the start of developing the PBK model. And then the interactions with the agency, we just basically use the model that we have. The model that we've developed now has complex absorption. Also, we verified the distribution, and we were able to predict the partitioning into adipose tissue and also verify that with available clinical data as well. But what you can do, but obviously, I mean, those types of, as I mentioned, often in these situations, it's hard to actually verify the exposures because we don't have clinical data available. But in this particular case, we did.
Thank you. I want to get your thoughts on the second question. Do you think a fold error range of 0.5 to 2 is acceptable? I think this is a very often asked question, and I really want to get your thoughts on this.
Yeah, so I think, this is similar to the earlier question, isn't it? Where, as I mentioned, I think this kind of range is acceptable early on. But when you're starting to make dose recommendations, you have to take into account the therapeutic window. So we would want to have a model that that captures the data accurately. I think the interesting thing going back to moxidectin, and also, so I'm gonna expand on question 1 and also talk about 2 here, is that when you actually, there is a very clear gender effect with moxidectin, because and it relates to the adipose tissue. When you go from male to females, the volume of distribution changes, and therefore, you have to think about the best fit of the model there as well. You're trying to optimize and capture the exposures in adipose tissue, because when you switch from male to female, the predicted volume can change significantly, and so can your actual profiles as well. And that's why I think it's important to have a model. You may end up going outside that 1.25 by trying to have a model that captures all the different scenarios. But as I said, again, I just want to mention that you have to put it in context of the therapeutic window.
Thank you. Maybe for the set of questions, we can look at the fourth one. So given moxidectin's pharmacological profile, did you consider using a facilitated dissociation model?
So we haven't used it for this, but so we haven't used a facilitated dissociation model, for the drug. But what we have done is actually predicted the binding based on the different albumin. Based on the different plasma protein levels for different populations to see what impact that has. And in fact, when you look at when you go from healthy subjects to subjects with onchocerciasis, that have different concentrations of plasma protein levels, it can have obviously a significant impact on the profile.
Thank you very much, Karen. I think the remaining questions from Henry, we will try to take them offline. There are a lot of interesting questions, but I think let us push these offline. Let me move to the question. Kenta here has a question. I'm sure you can see it. Maybe. So thanks a lot for sharing this very interesting and impactful case. What are the incentives, key drivers for re-evaluating infant doses for this drug, and how can we further incentivize others to follow? I think this is somehow related to the question I asked at the start. So what is that drive, the motivation, what are the incentives really to want to do this kind of analysis within this context?
Yeah, I think this is a really important question because I think that I think there are several things here. There's always the philosophical question about, about whether PBPK modeling can be used to, predict or optimize dose in special populations, and whether we are there yet with respect to the data. The PBPK models are data driven, but I think that the models we should be using them now. And even if we're not after a pinpoint accurate dose, what they can help us is to help support the clinical data that we have. So say for example this moxidectin, we've got the clinical study. We had that WHO safety recommendation. It was below 10, but how is that going to help us actually, make prescribing decisions about the drug? And I think this is what this example is showing, and also other emerging examples. We can use PBPK modeling to answer questions that we cannot answer using clinical study data, because we're conducting, a single dose study often single dose study in a small number of mothers. And, often we don't know when the samples were taken. Were they conducted in a malnourished population, healthy adult population? There are many different factors that can actually affect the exposure of a drug in milk. And therefore, I think that is where PBPK modeling really comes into play. There are many drugs on the market, many global health drugs where we have PBPK models, and we should be using these either to help inform initial doses, I think, for these types of studies, and also to support clinical lactation studies as well. Sorry, I'll put my soapbox away now.
Yes. Thank you. I would have liked
sorry,
to know,
sorry, I missed a key point.
This.
Oh, yes. Sorry, I just wanted to add to that. And and how can we incentivize? I think, as with all examples, it helps to disseminate examples. I think once we start sharing these examples, sharing publications, then it starts building a momentum. And I think that's how we can actually help incentivize others, sharing these case studies and also the publications. There are an increasing number of publications in this area where PBPK modeling has been applied in drug development and also the global health space. And I think we need to continue to do that.
Exactly. And I think it makes a difference if there's a concrete example that actually leads to label change. Yep. It makes a big difference. For now, I think we're going to have Shakira's question as the last. Were there any significant challenges or limitations encountered during the development of the model? I think this is a more general question from what every modeler's concern is usually. So, yeah. Over to you.
Yeah, so I think does this relate to moxidectin itself? I think with respect to moxidectin, and in fact, I think all models, is that we're, the models are as good as the data and that we're using to actually inform the models. So we need clinical data, but we also need in vitro data. One of the issues that we had with moxidectin, and also we see for a number of global health drugs, and I think also in drugs in development at the moment, is that many of these drugs are metabolically stable. They have very high log Ps or relatively high log Ps, and they're tight binders. And therefore, we need assays that are good enough in order to be able to generate the appropriate data to actually feed into these models. And for a number of the anti-malarial drugs in that publication that I showed, specific assays actually had to be designed to be able to get good estimates that could be actually used to generate PBPK models. So I would say that that is one of the key challenges. And as I said, I think the other as well is also the clinical data. Is in terms of the study population, especially if you're working on a global health drug. Obviously, moxidectin, it was developed, and the clinical lactation study was conducted in North America, but is obviously going to be mainly applied in in African countries, and also patients with onchocerciasis. So it's trying to marry up the different clinical sets of data and having relevant parameters that you can actually feed into your models from the populations as well.
Thank you very much, Karen. I think on that note, you've highlighted something. Still, you have some of these studies done in the global south, but a lot of the drugs actually use, rather in the global south, the other way around. The drug is studied widely in the US, and it's going to be used almost predominantly in Africa. So I think that goes into the direction of having the incentive to actually study these drugs additionally to what you already presented. I'm very sure if we had more time, we would discuss for an extra 1 or 2 hours, but unfortunately we don't have time, any time left. But on the same note, there are some open questions that I think are very important to follow up on. So I would really invite you to give us some of your thoughts on these questions. I think we can do this via email and then share this with the rest of the attendees. On this note, I want to bring this session to close. Thank you very much for attending, everyone. Thank you very much for the discussions. And once again, Karen, thank you a lot for sharing your expertise and the knowledge you have in this field. We really appreciate it. We will be communicating the topic of the next seminar series that is going to be scheduled at roughly 1 month from now. Thank you very much and have a good evening wherever you are. Bye-bye.
Okay. Thank you.